Okay, last time we're talking about the randomization, and we're talking about permutation test, and then on that basis, we're talking about the concepts of intention to treat. This concept is absolutely critical because the whole thinking behind current clinical trials is based on this permutation test and is based on intention to treat. And from a practical point of view, in addition to intention to treat, we will also need the treated set. And last time I mentioned about the fact that for a clinical trial, you can think of it as part of a prospective cohort study. And among all the prospective cohort studies, if you perform randomization, you're talking about clinical trials. So that's one way of looking at these two circles. Another way of looking at it is that for a clinical trial, when you receive the data after the clinical trial is over and blind is broken, when you look at all the data, there are three major data sets for each clinical trial. It's baseline demographic data, it's efficacy data, it's safety data. So randomization only is prepared for efficacy data. As to baseline demographic data or safety data, by nature, they are prospective cohort study. So they are observed in nature. So if you're looking at observed in nature, you have to look at treated set. Okay, treated set. So demographic and baseline data as well as safety data, they follow treated set. For efficacy, of course, ideally we follow intention to treat, but in practice, we may not be able to implement intention to treat. So we define something we call a full analysis set, FAS. And that follows the ITT principle. So that's the two fundamental principles of intention to treat are analyze all data and analyze as randomized. Okay. Now, we want to follow this principle. Report all data, analyze and randomize, but practically you can be implemented, and that has to be something more practical and feasible, and that's what we call a full analysis set, FAS. Now, for demographic data, baseline data, safety data, they follow treated set. Report all data, analyze as observed. Another data set is called PPS, per protocol set. We usually do not pay a lot of attention to per protocol set because per protocol set is a subset of intention to treat or full analysis set. Basically, if there are important protocol violations, if you exclude the protocol violations, then you'll come up with a per protocol set. Now, as we know, protocol violations are subjective. It's defined. Many of the per protocol set are defined post hoc after they see the data, and then they say, all right, this patient sort of took unallowed medication, so we have to exclude this patient, or that patient turn out to be not complying or not having the required inclusion and exclusion criteria, so we have to exclude these patients. And all these judgments are subjective. So proposal set is in general a lot less reliable than intention to treat. The reason I'm saying this is because everything is based on randomization. So if you cannot respect randomization, then the analysis could potentially introduce bias. So that's the important concept. We have to know, as I discussed, permutation test, we have to know intention to treat, and then we apply treated set in the baseline demographic or safety data. So these are important data sets. And proposal set is just for convenience, for other reasons, they come up with this proposal set, and it's usually more subjective than the other two sets. All right, so how big a difference can there be to have a intention to treat versus a protocol, per protocol set? All right, here is a slide we borrowed from the Rocket AF dataset. So in their analysis, when they look at the intention to treat data set, so this trial is trying to compare rivaroxaban against warfarin in a non-inferiority trial. So in this non-inferiority trial, if you look at the intention to treat, where they have 14,171 patients in this analysis, and they got the hazard ratio of 0.88 with a p-value of 0.117. That's the intention to treat. They made non-inferiority. All right, I think the non-inferiority margin for this trial was somewhere around 1. I don't remember exactly what the number is, but somewhere between 1 and 1.5. It's somewhere around here. So the intention to treat made non-inferiority. There's no doubt about it. But then they look at the per protocol set. They call it on treatment, the per protocol set. So if you look at the difference, 14,143 versus 14,171, there's a difference of only 28 patients. And it made superiority, right? Hazard ratio of 0.79 with a p-value of 0.015. So can you look at this from the intention to treat analysis or from the per protocol analysis, right? So the per protocol analysis, by and large, could be subjective. So from drug approval point of view, we should pay attention to intention to treat. All right, so I talked about the permutation test. We talked about randomization. So what does it mean by a scientific experiment? All right, a scientific experiment is defined by taking observations under controlled environment. So we know we can perform like biologic experiment, chemical experiment, physics experiment, and all these kind of experiments. They're all taking observations under controlled environment. For clinical trial, what is a controlled environment? You know, we're collecting data from live human beings. So human, they do anything they want. I mean, they have their behavior, they have their lifestyle, and all those kind of stuff. So what control do we have in clinical trial? The only control we have is randomization. So randomization is absolutely critical. So what we did in randomization was traditionally we did block randomization. Like I introduced previously, if you have a block of size 2, then for two patients, for any two patients, the first one may be randomized to A, the other would randomize to B, or vice versa. So that's a block size 2. Or you can do a block size 4. So for every 4 patients, 2 randomized to A, 2 randomized to B. It could be AA, BB, AB, AB, or BB, AA, all that kind of combinations. And then, in order to maintain balance, sometimes we have to do a stratified randomization. For example, if I run a trial with test drug against placebo, and let's say I want to have 100 patients in each treatment group, one thing I want to avoid is that it turned out in my test drug group, there are 60% male, 40% female, but in the placebo group, I got 40% male and 60% female. So in other words, there's some kind of gender imbalance. And I want to minimize that, all right? So how to minimize that? You do a stratified randomization. So the way to do a stratified randomization is that you randomize male patients against male patients, placebo, test drug kind of things. And you randomize female patients against female patients, test drug, placebo kind of stuff. So the way you do it is if we do block randomization, and if I'm doing block size 4, I will have a male block and a female block. So for the male block, the 4 male patients, 2 are going to be receiving placebo, 2 of them are going to receive test drug. For the female block, 2 of the female patients will receive test drug, 2 of the female patients will receive placebo. So that's how you do a block randomization with stratified by gender. Now, if I want to further stratify by age, I say age 65 and above, and age below 65. Now, I'm stratifying by gender, by age, so I'm going to issue 4 blocks, all right? 2 blocks for male patients, 2 blocks for female patients. Among the 2 male blocks, there's older patients, younger patients. Among the female blocks, there are older patients, younger patients. All right. Now, if I'm running a 4-week trial, and each patient will receive 4 bottles of test drug or placebo, so the double-blind medication. So when I do the drug shipment, if I have to randomize what's stratified by gender, stratified by age, now every time I need to send 4 blocks of drugs to each center, to each investigator or physician or clinic, all right? Now the 4 blocks, each block has 4 bottles, all right? For each, has 16 bottles, 4 bottles for each patient, all right? So now I ship out 64 bottles to this investigator so that hopefully they can recruit 16 subjects. All right. In the most balanced case, I would have 4 young males, 4 old males, 4 young females, and 4 old females. That'll be ideal. But of course, the reality is never like that. But at least in this way of randomization, I was able to hopefully maintain some level of balance, so that by the time I perform data analysis, the test drug and placebo patients, their male to female ratio are comparable, and their older and younger patients ratio are comparable. All right. So that's how you perform stratified randomization under block randomization. All right. So we say under block randomization, only a few stratification factors are practical, not too many levels with each factor. And minimization is a different way of performing randomization. Minimization allows for a balance in case of more stratification factors or more levels of each factor. So in the previous example I just mentioned, if you ship 64 bottles to this clinic, and by the end of the study, if the clinic does not recruit many patients, all right, suppose the investigator recruited only 3 patients, all right, and 2 of the patients complete the study, so they use 4 bottles of drug from each of them, and the 3rd patient dropped out after 2 weeks, so that patient took 2 bottles. So you ship 64 bottles of medication to the clinic, and only 10 bottles get used. The other 54 bottles are all wasted. So these are huge amount of waste of medication. All right. The history of clinical trial or drug development actually started from 1962. There's the Kefauver-Harris FDA amendment. So Kefauver and Harris are 2 congressmen. They sort of put together this act, this amendment, FDA amendment. They require clinical evidence for drug approval. And so from 1962, FDA started hiring statisticians, and then pharmaceutical companies started hiring statisticians. They started doing clinical trials, randomized controlled double-blind clinical trials, and randomization has been sort of block randomization. So block randomization is very efficient in a way of maintaining balance, because for every 2 patients, 1 on test drug, 1 on placebo. For every 4 patients, 2 on test drug, 2 on placebo. So you can maintain this level of balance. But then this kind of waste of drug based on block randomization, like I just described, under stratified randomization, became apparent. We'll get into the 70s and the 80s. So in the late 80s, there are statisticians publishing different ways of performing randomization, and they call it minimization. All right. So minimization allows for balance in case of more stratification factors or more levels of each factor. And here's how they do it. All right. Let's start with an example. In this example, we have 4 treatment groups. All right. Treatment groups A, B, C, and D. Now, here's how minimization does. Now, if I have a new patient coming in, all right, I have a new patient coming in. This computer algorithm will look at the existed number of patients randomized to each treatment group, all right? So this patient coming from center 11, and this is a young male patient, all right? Factor 1 is gender for male, female. Factor 2 is for age, old or young, all right? So now, center 11 came up with a patient. It's a young male patient, all right? Now, we need to randomize this patient. So what the computer does is the computer goes through this trial and look at how many patients have already been randomized. All right, so they look at how many patients from center 11 were randomized to A, B, C, or D. All right, there might be 2 patients randomized to A, 3 to B, 2 to C, 3 to D, or something, whatever the numbers already being randomized from center number 11. And then they look at all the male patients across centers, all right? So there may be 11 male patients in A, 9 patients using B, 12 patients using C, and 13 patients using D, or whatever, all right, across the whole study. And then they look at the young patients across the whole study, all right, across all centers within the study, across all genders, all right? Here, we're looking at center 11 across all genders, all ages. And here we're looking at the male patients across all centers, across all ages. And then we look at young patients across all centers, across all genders, and we got these numbers. Keep in mind that these numbers are not mutually exclusive, okay? I mean, these 2 are mutually exclusive. I mean, from the columns, they're mutually exclusive, but from rows, they're not mutually exclusive. But anyhow, you add up these numbers, you got X1, X2, X3, X4. All right, so I'm trying to explain how minimization randomized patients is that when a new patient come in from center 11, and this new patient is a young male patient, how does minimization randomize this patient? All right, so minimization says, all right, this is center 11, young male patient, and we calculate X1, X2, X3, X4 based on all the patients already randomized in the study. So I got these numbers. Now, here's how minimization does. It first try to come up with 2 sets of treatment groups. Remember, we have group A, B, C, and D. And the first thing they do is to look at set M and set N. All right. So we have A, B, C, and D, the 4 treatment groups. All right. Set M may be A, B, and C, and set M being D, or set M being A, and set M being B, C, and D, or whatever. All right. Now, the way they define the treatment set is this. All right. M is a set of treatment groups J such that Xj is minimum. All right. So we have X1, X2, X3, X4. Among the 4 numbers, there's always a minimum number. All right. If the minimum number applies to all 4 groups, and all 4 treatment groups are in M, and N is going to be an empty set. If there's only one minimum number, then M contains only one group, and N contains the other 3 groups. Okay. So M is a set of treatment groups such that Xj is minimum, and N is a set of the other treatment groups. So that's how they define these 2 treatment groups. Now, the example here is, suppose that in the above example from here, we have X1, X2, X3, X4, and we got X1 equal to 12, X2 equal to 11, X3 equal to 11, X4 equal to 13, then M contains treatment groups B and C, and N contains treatment groups A and D. Okay, so we have 4 numbers. So the way perform randomization is to look at how many patients already being randomized from center 11, how many patients being randomized of the male, how many male patients being randomized, and how many young patients being randomized. And you got these numbers, you add them up, you got X1, X2, X3, X4, and among the 4 numbers, you have a minimum number. So you look at the minimum number, and X2 and X3 have the minimum number, so these 2 groups go to M. So set M has a minimum, which is group B and group C. And N has group A and group D. Okay. So this is how the minimization is performing the randomization. All right. Now, randomness was then introduced at 2 different levels. A first random variable dictated the choices between M and N. So if you look at it this way, this is a kind of imbalanced coin randomization. All right. You can set up at the beginning of the randomization program to say the probability randomized to M is probably 0.55. And then the probability randomized to N will be 0.45. All right. Now M, because there are fewer patients randomized to M, so you want to allow a little bit more probability for this new patient being randomized into. All right. N already has more numbers, so you have less probability this new patient will be randomized to A or D. So this patient is more likely to be randomized to B or C. Okay, that's how you set it up. So that's the first randomization. The second randomization, the second one dictated the choice of a treatment group within the chosen set. All right. So now, if you come up with a random number, you said this patient will be randomized into M, then the next step, you do another randomization to allocate this patient into B or C, that kind of things. So that's how the minimization is performed. The minimization is sort of a different randomization technique after the block randomization because of the way we try to minimize the waste from the block randomization. All right, so that's the basic principle about minimization. All right, here I'm going to introduce an example of based on permutation test. Okay, now in permutation test, here is an example I did. Well, almost 20 years ago, so after turning into the 21st century, we have a drug. We filed NDA and we're preparing for the advisory committee meeting. And one of the things we're concerned about is because in that trial we used a permutation test. I mean, we used a minimization to perform randomization. So we're concerned if there will be in the advisory committee meeting, there will be questions about the randomization. Instead of block randomization, we use minimization. Will we be challenged? So we prepared the following analysis. And the trial was 2 doses against placebo, and the primary endpoint is a binary. So for that drug, our primary endpoint is responder or non-responder. So a patient may respond to study medication or may not. All right. So we use Cockle-Mendel-Hansel test. All right. So the pairwise comparison are high dose versus placebo and low dose versus placebo. Now, the CMH test, all right, we're using a chi-square test with 1 degree freedom. I think chi-square test with 1 degree freedom, alpha being 0.05, I think the critical point is like 3.84 or something like that. And we got a chi-square value of 6.5534. That's pretty good, all right? And the low dose against placebo, we got 8.5349, all right? So we got statistical significance in both doses, all right? And then, of course, if you look up the chi-square table, you got the p-value of 0.0105, all right? So you sort of do the numerical integration or whatever, you can get this p-value for corresponding to a chi-square test value of 6.55, the p-value is 0.0105, corresponding to a chi-square value with 1 degree freedom of 8.5349, you got corresponding p-value 0.035. So that's the asymptotic p-value. So what we did was that we did a permutation test was 10,000, was 10 million permutation. All right, so here's how we did the permutation. Remember, there's 3 treatment groups, they're high dose, low dose, and placebo. So when we do the permutation test here, what we did is that we exclude the low dose group. So we have only high dose and placebo, and we re-randomize using minimization. Once we re-randomize, we calculate a chi-square value. And then we re-randomize again, calculate another chi-square value. Re-randomize again, calculate another chi-square value. So we did 10 million times. So there are 10 million re-randomization. And so you got 10 million chi-square values. And then with that 10 million chi-square values, we look at where does 6.5534 located. And you look at to the right of 6.334, you count how many chi-square values we obtained from the permutation. And then divide that by 10 million, we got 0.0054. And we did the same thing for the low dose versus placebo. And then you look at the 8.5349 among the 10 million permutations and look at how many of them are greater than this value. You got a p-value. I mean, you count the number divided by 10 million, you got 0.0066. And here's what we obtained. I mean, if you do a histogram of the 10 million high dose versus placebo, all right, you did 10 million of them. I think this is high dose, right? Yeah, high dose versus placebo got 6.55. Here's 6.55, right? And we did 10 million of them. Here are the number of calculated chi-square from the 10 million permutation. And you count to the right of 6.335, 6.5534, we got sort of 0.0054. So that's the number over here divided by 10 million, you got this p-value. And then for the low dose, we look at this cut and we got a p-value of 0.0066. All right. So that's an example of how you apply minimization in real world situation. You not only get the observed chi-square, you can perform the traditional way of table lookup and things like that, get your p-value. Or you can do a permutation test, you got another set of p-values. So I want to come back and say a clinical trial is double-blind, randomized, controlled clinical trial, right? There's three properties: it's randomization, double-blind, and controlled. And randomization is the entire key of clinical trial. All right, the whole idea of randomization came from R.A. Fisher. All right, so remember earlier we talked about lady tasting tea example. Actually, the professor that proposed this test is R.A. Fisher. And of course, nowadays that test is known as Fisher's exact test. He also proposes permutation test. All right, you can do permutation and you can get a hypothesis test. This entire concept built up a framework of statistical hypothesis testing. This framework of statistical hypothesis testing transforms the subjective feeling into objective probability. So it's not that I'm feeling better, but we will have a probability that the drug does not work. All right, because we always set up this null hypothesis, where the null hypothesis says mu t equal to mu p, which is test drug mean and placebo mean is the same, or we cannot differentiate them. But now, if you have a statistical hypothesis test and you run clinical trial and you see patients' response and you were able to separate two distributions, all right, there are two distributions, or test drug distribution and placebo distribution. If you can separate them, if you can separate these two distributions, and then you can say, I reject the null hypothesis, not by saying the null hypothesis is not right, but to say, if the null hypothesis is true, the probability of observe this phenomena is very low. So how low? It is below alpha. And then FDA can approve the drug on this basis, not saying the drug works, but by saying the probability the drug does not work is controlled under alpha. All right. So this is a major breakthrough. It's a scientific, it's one of the most important scientific contributions in 20th century, that we were able to use the statistical hypothesis test to approve drugs. And these data actually extended human life expectancy and largely improved the patient's quality of life. All right. So everything is based on the permutation test. And Fisher demonstrates that the permutation test can be approximated by the asymptotic test. So the permutation test is a foundation. But the asymptotic test that we use today now, z-test, t-test, f-test, chi-square test, it can be acceptable is because it it asymptotically approximates the permutation test. All right. So still on randomization, we sort of start with block randomization. Everybody knows how to perform block randomization. You know, block size 2, size 4, or whatever, right? You can perform block randomization. But then we introduce this minimization. Now, once there's minimization, the key now, the randomization is no longer static, it's dynamic. What do I mean by static? If I do a block randomization, I can set up a randomization list, all right, ahead of time before I start the clinical trial. I can have this whole list of patient 1 is A, patient 2 is B, patient 3 is B, patient 4 is A, whatever. I can have this list of the randomized patients before the patients are actually entering the study. But with minimization, you cannot do that because minimization is performed based on the number of patients being randomized to each treatment group before this new patients enter the study. So it's not as it's a dynamic kind of randomization. And so nowadays there are contract research organizations we call CRO, contract research organizations, that specialize in performing randomization. And nowadays we call them IRT, all right, interactive response technology. All right, let's see how they did it. All right. Suppose in a 4-week study, patients are randomized to 4 treatment groups, weekly outpatient visits, and each patient takes weekly supply of study drug at each visit under block randomization. 4 patients are needed to complete a block. If we do a block size 4, then patients may drop out of the study for various reasons. Prepared drug supply cannot be used. It's high waste of drug products. So we talk about that when we'll talk about minimization. Now, what the IRT does is that the clinical research coordinator calls the phone. So what happened is that when a new patient comes in the clinic, so in the clinic, there's a clinical research coordinator, usually a study nurse or physician assistant or somebody who sort of oversees this clinical trial and sort of recruiting patients and checking the conditions for randomization, stuff like that. So now, if a new patient comes in, and by going through the screening, by going through the baseline, everything like that, it's time to randomize. So at the time of randomization, the clinical research coordinator calls the phone. This IRT, there's a phone number, they call the phone. And once the computer picks up the phone, the computer performs 2 levels of randomization. There's patient level randomization and there's bottle level randomization. All right, so what it actually did is if we agree to use IRT, so we work with this CRO to say, okay, you're going to perform IRT for us. And then the drug company's pharmacy will work with the IRT team. So the IRT will issue the sort of the bottle numbers, all right? So you can probably have maybe 1 million numbers, all right? It's just numbers. You can do a sequential numbers to say, if this study needs, say, 200 patients and 4 bottles per patient, I'll need 800 bottles, but I may need some oversupply. Maybe I need 1,000 or 1,100 bottles. All right. So if I have 5 digits, I got more than enough. All right. So 5 digits to cover like 1,200 bottles. That's not a problem. So the IRT, the CRO, will provide all these bottle numbers to the company. So the pharmacy within the company will receive these bottle numbers, and then they'll prepare drugs according to these bottle numbers. All right, maybe every first 10 bottles being placebo, next 10 bottles being test drug. So every 10 bottles, there's certain kind of way of arranging so that the pharmacy knows which bottle has what drug, and they have to do the QC and everything, make sure the bottle has what it's supposed to have. Now, when there's drug shipment, the IRT, or this contract research organization, will notify the pharmacy to send maybe 20 bottles to clinic number 1 or investigator number 1 or center number 1, 20 bottles to here. And they give pharmacy the bottle numbers you need to ship. All right, so these 20 bottles go to center number 1, the next 20 bottles go to center number 2, the rest, another 20 bottles go to center number 3, and so on and so forth. And the CRO who runs IRT, the computer knows exactly which bottle has what and which bottle goes to which clinic. So suppose bottle, let's say 17100 to bottle 17200. We have all these 100 bottles or whatever goes to center number 1. Now, when the study nurse called IRT, all right, the IRT will start asking questions. This phone number is a computer system. Will ask the nurse which protocol you're calling from, all right, which protocol you're calling about, and which center you're coming from. And is this a first randomization, or this is a resupply of previous randomization? And so the study nurse or the coordinator will answer all these questions using the key on the phone. And of course, the system will ask about the patient's date of birth to determine the age, to ask the patient's gender, and all those kind of stuff. So the computer will receive all these information and do the randomization. And then the computer will tell the CRC, clinical research coordinator, say, bottle number 17131. All right, that is whatever placebo test drug being already prepared, already shipped to the center. And the CRC will dispense this bottle to that patient. All right, so the randomization does this. They do a patient level randomization and they do a bottle randomization. So it's all randomized in bottles, and the bottle numbers are all random, allocated. All right, and then the of course in a certain way that the company's pharmacy knows which bottle has what, and then the IRT will help the CRC to dispense this bottle to the patient. So that's pretty much randomization, all right? And I want to focus the two lectures, the previous lecture and today's lecture, on the 3 fundamental properties of clinical trial: randomized, controlled, double-blind. So I think I have a brief introduction of randomization. The next few slides, we're going to talk about controls and double-blind. Basically, there's 3 types of controls: placebo control, active control, and historical control. Okay? Placebo control is gold standard. If you can differentiate the 2 distributions, right? If you can differentiate the 2 distributions, test drug versus placebo, if you can separate these 2 distributions, you're able to achieve the objective to demonstrate that the probability the test drug is different from placebo, or the probability the test drug is another placebo is controlled under alpha. You can do that. All right. Now, active control is a different setting. All right. Active control is a drug already approved and already on the market. Okay. So in many situations, when the test drug compare against the active control, if the active control is already approved, we may not be able to show superiority to the active control. So in many of the active control trials, we're talking about non-inferiority trials. So what do I mean? Let me give you an example. Antibiotics, right? Before there's penicillin, if patient got infected, patient may die, patient may suffer, and all that kind of situation, but there's no antibiotic to help them. But once there's penicillin, once there's other antibiotics, now you can control these kind of infection agent, and patients responded to antibiotic. So when penicillin was ready, there was no placebo-controlled trial. We just know penicillin worked, all right? Now, if without any antibiotic, the patient response rate may be only 30% or something like that. Well, in today's world, most antibiotics achieve like 85% or 90% response rate. Now, if you compare 85% against 30%, it's 55% improvement. Now, you want to come up with new antibiotic, and you want to compare with the existing antibiotic. You want to provide 50% of improvement? No way. All right. So you're comparing to this approved antibiotic using a non-inferiority trial. You hope it's not worse than the existing antibiotic. And of course, we need newer antibiotics because of resistance and the other things, right? We need more or newer antibiotics. But for antibiotic development, it's not ethical to run placebo-controlled trials. So you have to do active control trial, you do non-inferiority trials. And ICH-E10 is specifically about how to select active control, and they talk about the non-inferiority trials. Historical control is the weakest evidence. So what is historical control? If you do a single group trial, all right, I only have test drug. Now, how are you going to compare? So I'm going to compare with published literature and stuff like that. I'm going to compare with the past history kind of stuff. So that's the weakest evidence. The strongest evidence is placebo-controlled trials. Now, the next level will be active control trials. All right. If in active control, you can still show superiority, then it's, again, gold standard. But in most active control trials, we can only do non-inferiority, so that's a secondary choice. And historical control trials, the weakest evidence. And there are times you have study designs with both placebo and active control trials. Now, here I want to give you a few examples. All right. The use of active control, sometimes the active control could change over time. All right. And here I'm talking about Vioxx as an example. All right. Vioxx was discovered and developed by Merck. And Vioxx was, I think, approved in 1999. And in 2004, Merck withdrew Vioxx. In 2004, Vioxx is known as COX-2 inhibitor. After 1999, there are many COX-2 inhibitors. I think most of the major pharmaceutical companies, they were developing COX-2 inhibitors. In September of 2004, Merck withdrew Vioxx from the market. And when Merck withdraw, you cannot use Vioxx as active control. All right. So I remember we designed a trial using Vioxx as active control. And in September, when they withdraw, we had to immediately stop the Vioxx treatment group, and we let the other treatment groups continue. All right. So there are many situations the active control could change. All right. And of course, we have to worry about the dose and regimen of active control when we select these active controls. Now, before getting into blinding, I just want to give you an example about expiration dates. All right. I remember there was once we were running a clinical trial against a market leader. And we went to this market leader and we went to the company to say, we want to buy your drug. And usually we not only buy their drug, we buy their placebo also. Right? We say, we want to buy your drug. and they say, we're not going to sell it to you. All right, you want to compare it to our drug, you can go to the pharmacy and get it. And yes, we did. We went to the pharmacy and got these drugs. But there's problems blinding. All right, suppose our test drug is tablet and this comparator is capsule. So how can you blind? How can you perform double blind? All right. So we did this double dummy, double blind. In other words, we're going to have our test drug being tablets. So we're going to have placebo tablets of our drug. And their drug is capsule. We're going to have placebo capsule. So each patient is taking one tablet and one capsule. For patients randomized to our test drug, they'll take the 2 test drug tablet, and they take a placebo capsule. For patients who randomized to the active comparator, they're going to take a 2 capsule of the active control, and they take a placebo tablet of our test drug. So that's what we call double dummy, double blind. Now, when we did this from the local pharmacy, so we try to go to this pharmaceutical company and try to buy their active control. They don't sell us, so we go to the pharmacy and we try to sort of do the blinding, do the trial. The biggest problem here is expiration date, because you buy the drug from the original company, you usually have like 18 months or 18 months or 24 months shelf life. But if you go to a pharmacy, you only get 3 months shelf life. So we have to do a lot of resupply. It's a lot of operational headache. And there's another situation that, I mean, the placebo for active control is another headache. And that time, what we did is that we bought their capsules, but the capsules have their logo on it. So how can we blind them? So what we actually did is to over-capsulize. In other words, outside of their capsule, we added another capsule. So we prepared the placebo, and we have this over-capsulized capsules. We have another capsule on the outside of their capsule to blind their drug label or their drug logo. Okay, and so we were able to successfully demonstrate our test drug is non-inferior to that capsule, the active control capsule, and we submitted to FDA. And FDA says, wait a minute, you over-capsulized their capsule. How do you know the properties of their active control is not changed? So we had to come back and do a bioequivalence study to show over-capsulization does not change their PK. All right, so that's control. Now finally, we'll talk about blind. All right, let me give you an example. All right, if in a dose response study with placebo, 5 milligram, 10 milligram, and 20 milligram, and we only have 5 milligram, 10 milligram tablets, and our 5 milligram, 10 milligram tablets look different, how do you do the double blind? So we have to do 3 bottles. All right, bottle 1 contains 5 milligram or 5 milligram test drug or 5 milligram placebo. Bottle 2 and 3 having 10 milligram tablets or 10 milligram placebo. And if a patient is randomized to placebo, that patient will take all 3 bottles of placebo. All right. Now, if a patient is randomized to 5 milligram, that patient will take the 5 milligram true drug and placebo from bottles 2 and 3. If patient randomized to 10 milligram, that patient will take 10 milligram from bottle 2 and placebo from bottles 1 and 3. If patient randomized to 20 milligram, they take placebo from bottle 1 and 10 milligram from bottle 2 and 3. All right. So blinding is not a slam dunk. All right. Blinding, as I mentioned, there's double dummy, double blind, and there's different shape of the test drug or different formulation of the test drug. You have to consider the blinding feature for that test drug to run clinical trials. All right. Now, let me give you one more example about a complicated blinding procedure. So I work on Macugen. Macugen was developed for age-related macular degeneration. All right. It's one injection to the eye. So we have two eyes. One is study eye, the other eye is fellow eye. So the patient will have AMD on one of the eyes, and we have to inject the Macugen into the eye. All right, it's a sham control. So we're going to have evaluation physician and injection physician. So the evaluation physician does the evaluation for the patient and the primary, secondary endpoint, and all those kind of stuff. And the evaluation physician is blinded, but the injection physician is not blinded. So what they did is the injection physician will have to prepare, you have to do the sterilization and all those preparation procedure for the study eye. All right. Now, if your study eye is a left eye, it was instructed to look at the right side, and then the needle will come from the left hand side. If the study eye is the right eye, the patient is instructed to look at the left, and then the needle will go into from the right. Now, for a sham patients, there's no actual injection. There's all the same preparation, but the injection physician will just touch on the eyeball, but do not actually inject into it. Blinding active controls with double blind, double dummy, double blind, and sometimes different frequency, different route administration can be very complicated at times, and there are ethical concerns. So now I've introduced the three properties of a clinical trial. There's randomization, control, and double blind. And of course, there's parallel studies. Most phase 2 and 3 are studies of parallel studies. You randomize the patient to test drug or placebo. The patient on test drug will take test drug for the whole time. If the patient is randomized to placebo, the patient will take the placebo all the time. So these are parallel studies. So patients randomized to a treatment group stays in the same treatment group throughout the entire period. All right. Crossover trial is different. Subjects randomized to sequences 1 or 2. Sequence 1 could be receiving A first and receiving B. Sequence 2 may be receiving B first and receiving A. So a patient randomized to sequence A, then the patient first receive A. If a patient randomized to sequence 1, the patient first receive treatment A. Then after a washout, receive treatment B. If patients randomized to sequence 2, that patient will receive drug B first, and then after a washout, receive treatment A. So there's a washout between 2 periods. So we can do a 2-period, 2-treatment design as 2x2, and there can be more complicated 2x4, 4x4, 2x3 kind of stuff. Crossover designs are more common in phase 1, mostly in bioequivalence and bioavailability studies. All right, especially in bioavailability design, bioequivalence study design. Not appropriate for long-term studies. Crossover, if you're doing a 1-year study, once you cross over, become 2 years. All right. It sort of eliminates the inter-subject variabilities and requires smaller sample size. So there are benefits of each type of studies. The process of clinical trial usually start with a clinical question, and they design the protocol. And then there's study design, pre-specified analysis, SAP, and then the monitoring. After the study is completed, you perform analysis reporting, you perform a clinical trial report, a clinical study report. So study design is absolutely critical, all right? For the rest of my lectures, we're gonna pay a lot of attention about study design issues. Monitoring, statistics and involvement monitoring are not as much as the design or analysis, but there's also data monitoring. That's another topic. Okay. And analysis reporting, after the study is completed, you unblind and then you perform analysis. So I think we can stop here.